So today we're going to look at the lymphatic and immune system. So these two systems are very tied together. The lymphatic system is also tied in with the circulatory system because the fluid is going to be continuous with the blood. It is going to be the site of a lot of your immune function. So our human body is going to recognize anything other than its own tissue as an invader. And so when you have these foreign invaders in the body and they're capable of living in there, we consider it a pathogen. This diagram here just shows you some of your lymphatic tissues and organs in the body, as well as the layout of lymphatic vessels. So your plasma is going to contain antibodies against cells that are not your own. These are formed when an infant is about 3 years old. So you're going to have a lymphatic system in the body that's going to help to identify and destroy these foreign invaders. And it does this through antibodies. So when you have a reaction against a specific foreign body, we call it an immune response or immunity. So we have two major types of immunity. Innate or nonspecific immunity is what you're present with at birth. It may be something about being human that's not compatible with that pathogen, but it doesn't involve specifically recognizing the invaders. There's no memory component to it. You have first and second line of defenses that their whole goal is to try and keep the invader from getting into your body, and its response isn't to specific invaders. It's going to have the same general response for anything that's foreign. Adaptive or specific immunity, on the other hand, is going to involve recognizing the specific invader. It can have a memory component, and then that immune response is going to be adapted and specialized to that specific invader that comes in. So your immune system is going to include all the parts of the body that help with this recognition and destruction. White blood cells, phagocytes, lymphocytes, bone marrow, lymph nodes, tonsils, thymus, spleen, all part of it. So when a person is susceptible, we say that they lack resistance. So they're able to be infected. So when we look at this system, it's a transport or conduction system. You're going to have the lymphocytes and lymphatic vessels that are going to be involved with that aspect of it. But we also have the production organs that are going to actually create some of the things, and those would be your lymph nodes, tonsil, spleen, and thymus gland. Particularly the thymus gland for T cells. So when we look at the functions, you're going to have fluid balance, which is also going to involve returning the lost plasma back to the bloodstream, fat absorption, and defense. So your circulating blood will release about 30 liters of fluid a day into the interstitial space. You can't just leave it there or you would swell up. So of that, 27 liters are returned back to the circulation. There's about 3 that stay in the interstitial space. And it's constantly shifting through. It's not the same 3 liters all day long. The lymph is going to pass through the lymphatic system and go back into the blood vessels. So in addition to water, your lymph is going to have some of the plasma proteins and other substances extracted from cells. So because we can't leave it all in the tissues, that would cause miserable swelling. You are going to return lost plasma proteins into the bloodstream, and that goes with the liquid. Again, this is another picture here showing lymphatic vessels, as well as some of your lymphatic organs and tissues. Fat absorption occurs in the lymphatic system. So things like your dietary lipids and the fat-soluble vitamins A, D, E, and K. And then you've got defense. Your immune system will be involved with it. Lymphocytes, which will be your B and T cells. So fat absorption, this is going to occur in the small intestine, and then they get transported into the venous circulation. And different fats are absorbed different ways. So when we look at the components, your vessels begin as lymphatic capillaries that are closed at one end. So with your circulatory system, you have a vessel that's going to carry blood to a location and a vessel that's going to carry it away. With the lymphatics, you only carry things away. So These lymphatic capillaries are going to unite to form your larger lymphatic vessels. They are more similar to the veins in structure than they are in the arteries, but they've got even thinner walls and more valves. And on the way back, they're going to pass through lymph nodes. They're encapsulated organs that are going to have aggregates of B and T cells in there. And then you've got lymph nodes towards the large veins and above the heart. So your lymphatic drainage is not symmetrical. I like this picture here with the green and yellow body. So the area that is going to go into the thoracic duct is in the green. It would be the left arm, part of the head, the legs, and part of the torso. And then your right lymphatic duct is going to be the area that's in the yellowish color, be the right arm, part of the torso, part of the head. The exact cutoff is not going to be a sharp straight line like this. There's a little bit of variation in there. So the lymph begins in the lymphatic capillaries and it's made from the interstitial fluid. It's the same fluid, it just changes names when it gets into the capillaries. And then it's called lymph when it's in the lymphatic vessels. It will eventually be emptied into the bloodstream and join the plasma. So the lymph has less protein than the plasma. So your lymphatic capillaries are found throughout the body, except in the avascular tissues, the CNS, portions of the spleen, and the red marrow. But they are pretty much everywhere else. So they start as closed-end tubes, and they're found in the spaces between the cells. So they are a little bit larger diameter than the blood capillaries, but they've got a one-way structure. So you'll have overlapping endothelial cells that will open when the tissue pressure is high, creating one-way valves. This will allow the interstitial fluid to flow in, but not out. Then we have anchoring filaments. These are going to pull openings wider when the interstitial fluid accumulates. This is going to keep the tube from collapsing when the outside pressure builds up. And then in the small intestine, your lymphatic vessel is called a lacteal in there that will be specific for absorbing dietary lipids. Chyle is lymph mixed with the lipids. So this picture here shows how you have lymphatic vessels, arteries, and veins all intertwined together that are in the different types of areas. So you've got the venous system, tissue cells, lymph capillaries, and so you can have the fluid going into the lymph capillaries. So what's going to make the lymphatics different from the venous system is particularly the lymph nodes. So there isn't anything that's going to filter in the venous system like there is with the lymphatics. So your lymphatic vessels are going to unite and they're going to form lymphatic trunks. So you'll have the lumbar, intestinal, bronchomediastinal, subclavian, and jugular. So the lymph will pass from the lymph trunks into the two main channels, your thoracic duct and the right lymphatic duct. So it passes from the ducts into the venous blood and it will enter back in the subclavian veins. So your left subclavian vein is going to receive lymph from the left axillary vein, the lumbar trunk, jugular trunk, and the thoracic trunk. So this is another diagram here showing the drainage from the body into the lymphatic vessels. So here you've got the lymphatic duct. Things are going to go to the lymph node, lymphatic capillaries. And this just shows here where these are going to come in. So this is going to be the right side and then the left side in there. So here's another picture that shows the area drained by the right lymphatic duct and the left thoracic duct. So we have Starling's law coming back again. It's going to be similar to Starling's law for venous return. So when you have low pressure, you have lower speed. The valves are important to prevent backflow for the same reason. There is no pump or rhythmic contraction of the vessels. So what you're going to have to rely on is the skeletal muscle pump, the respiratory pump, and the thoracic pump. The heart isn't going to do it this time. So the skeletal muscle pump is going to work similar with the milking action. Same thing with the respiratory pump, with having the lungs expand during breathing for your thoracic pump. And then you'll have rapid flow of blood in the subclavian veins. So we have diffuse aggregates of lymphocytes and macrophages, especially in the passages that are open to the exterior. So if it's open to the exterior, that leaves the potential for invaders to get in, and that's why we want to protect those areas a little bit more. So this would be involved with your respiratory, digestive, urinary, and reproductive systems. So we group them into two groups based on their functions. So your primary lymphatic organs are going to provide an environment for the stem cells to divide and mature and basically hang out to become immunocompetent. So this would be the red marrow and the thymus. So the red marrow gives rise to the B cells. Thymus is going to give rise to the T cells. The secondary lymphatic organs, lymph nodes, spleen, lymphatic nodules, they're a place for the lymphocytes to hang out and to have the invaders get caught. So your thymus is going to shrink as you become an adult. Starts out being 70 grams in an infant, and then it will shrink down to 3 grams as an adult. So it's located in your mediastinum, and it's going to deteriorate after puberty, and eventually you'll have diminishing effectiveness of the immune system as you get older. So we divide up the thymus into 2 thymic lobes. They're divided by septa into smaller lobules. It's going to have a dense outer cortex and then a pale central medulla. So the thymosins are the hormones that are going to promote the development of the lymphocytes. They'll divide in the cortex, and then the T cells will migrate into the medulla. So if you think of it as T cells or thymocytes, they're called that because of their development in the thymus. It makes it a little bit easier to remember. Mature T cells leave the thymus through the medullary blood vessels. So you've got reticular epithelial cells in the cortex that are surrounded by lymphocytes in the cortex. This is going to help to maintain the blood-thymus barrier. So when you secrete the thymic hormones, they're going to stimulate stem cell division, T cell differentiation, reticular epithelial cells in the medulla to form the concentric layers. We call them the Hassel's corpuscles. And then the medulla has no blood. So there's no blood-thymus barrier. The T cells can enter or leave the bloodstream here. So this is showing here a tissue sample of a thymus here that is purple, and you can see where you've got the medulla in the center. You've got the Hassel's corpuscle. And then this shows where your thymus would be located in the mediastinum. And this is showing it a little bit closer. So lymph nodes are located along the lymphatic vessels, and you'll see these throughout the body except in the brain. So they can be a site of cancer growth and metastasis. So things that your lymph nodes are going to do, they're going to filter out potential harmful species from the lymph. So one of the things that can be an indication of being sick is swollen lymph nodes because the filter is being activated. They're also going to provide immune surveillance with the macrophages and lymphocytes, and then an area of lymphocyte production. The stroma is the supporting connective tissue. Around the outside, you've got the capsule, and then you've got the trabeculae in between the reticular fibers and fibroblasts. So if we come back to this picture here, you can see the pink lines are the trabeculae. So the parenchyma is the functional part. You've got the outer cortex that's going to have aggregates of B cells and lymphatic nodules in there. Sometimes referred to as the follicles. This is going to be where you're going to have plasma cells and memory B cells form. Then in the inner cortex, it's mainly T cells and dendritic cells. And then in the medulla, you have the B cells, and then you'll have antibody-producing plasma cells from the cortex and macrophages and some follicles. So this allows you to see here again the layout of a lymph node here, where your reticular fibers in here, you've got the medulla on the inside, the cortex is the outer portion. And then there's more pictures here that are even larger. So again, like with any of the notes, take what you like for the pictures, and you can ignore the other ones. There are plenty of duplicates in there. So the spleen is the largest lymphoid organ. When you look at it, it actually is going to look very similar to a very large lymph node. It's going to filter the blood rather than the lymph, though. That's the difference. So it will detect and respond to foreign substances that are in the bloodstream. It's going to help with the destruction of the worn out red blood cells. So we talked about that back in the blood. And then it can act as a blood reservoir, just like the liver can. So the white pulp is going to be the lymphatic tissue that's around the internal arteries. So lymphocytes are going to be stimulated here. The red pulp is the fibrous network of the macrophages and red blood cells. And these will have your capillaries connected to the veins. So your macrophages are going to be part of that recycling. They're going to remove the worn out cells and invaders. The spleen will actually store about a third of the body's platelet supply. And then the production of the red blood cells during the fetal life will occur here. Only the lymphocytes are produced in the spleen as an adult. So you get more blood production in a fetus there. So the lymphatic nodules, they are not surrounded by a capsule. They're going to be scattered throughout the lamina propria, the mucous membranes, and then lining the gastrointestinal, urinary, and reproductive tracts. So we do have some smaller lymphatic nodules. We call them the mucosa-associated lymphatic tissues. They're usually small and solitary. And then you have some that are larger, like your tonsils in your mouth and throat, the Peyer's patches in the small intestine, and then the appendix. So the appendix actually does have a function. So here you can see Peyer's patches, appendix, colon follicles. So with your tonsils, so for a certain generation, your tonsils were removed pretty regularly in people. They did it if you got sick very often at all. And they no longer do that because they have a better understanding of what their purpose is, that they actually do play an important role in the immune system. They are the ones that are most likely sampling the pathogens that pass through the mouth and the upper airway. So if they can get caught here, they can be removed without consequences. You remove it though, and now you've taken out one of the big filters. There's three sets of these. The pharyngeal tonsils are adenoids. They're going to be the higher ones. The palatine tonsils go closer to the palate, and then the lingual tonsils by the tongue. So here you can see in the mouth. Again, those lymphatic tissues. So when you are going to have your leukocytes be derived, we start in the red marrow. You have the hemocytoblasts that will form your lymphoid stem cells. So with these lymphoid stem cells, some will migrate to the thymus and produce your T cells. You can have other lymphoid stem cells that are going to become your B cells and natural killer cells, and those will go into the peripheral tissues for the immune responses. So again, this is another picture showing it a different way. You have the stem cells in the red marrow to give you your undifferentiated lymphocytes. Some are going to proceed to the thymus to become T cells. Some of the undifferentiated lymphocytes are processed and become B cells. And then both the B and T cells will travel through the blood to the lymphatic organs and lymphatic tissues. So in terms of microorganisms in the body, there are a lot of them. There are more bacterial cells in your body than there are human cells in your entire body. There's more bacterial cells in your gut than human cells in your entire body. So you have a lot of them. They're constantly going to be exposed to food, water, air, etc. So when we look at our resistance to microorganisms, we have some non-specific resistance to disease. They're general defensive mechanisms. So they will be effective against a wide range of pathogens or microbes that can cause disease. And their response is always the same. So there's no memory, where your specific resistance is able to fight a specific pathogen. This happens after exposure, and it's a slower response to happen. So this will include your cell-mediated immunity and antibody-mediated immunity. And this is where you would form your immunologic memory. So just the species you are gives you some inborn resistance. So there are some things that don't want to live in humans. You're naturally going to be immune to those things. So this gives us a way of being able to kind of visualize the groupings of the immune. For innate immunity, you've got your first line of defense and second line of defense. Your first line of defense would include skin, mucous membranes and their secretions, and the normal microbiota. Second line of defense, some of this non-specific immune cells like phagocytes, neutrophils, eosinophils, dendritic cells, macrophages, the processes of inflammation, fever, and other antimicrobial substances. The third line of defense, or adaptive immunity, this is where you're going to have your specialized lymphocytes, T cells, and B cells, and form your antibodies. So looking at your first line of defense, this is largely going to involve the skin and mucous membranes. They're going to give you both physical and chemical barriers. So physical barriers are just that. It would include the epidermis. You have closely packed keratinized cells. They are periodically shed off. Your mucous membranes, they help to trap the microbes and foreign substances. Nose hairs, they help to trap and filter. The cilia of the upper respiratory tract will help to trap particles and then move them up and out. Fluids catch things, and then the lacrimal apparatus of the eye. You have the constant washing of the tears so it can mechanically flush things out. Then we have chemical barriers. So things like your sebaceous glands. Sebum is a protective protective acidic film. Perspiration, gastric juice, vaginal secretions, they're all slightly acidic. Lysozyme helps to break down bacterial cell walls. So you have some of this in saliva, perspiration, nasal secretions, and your tissue fluids. Saliva to help wash out the mouth. The urinary system is going to help flush things out with the urine. And then defecation and vomiting if your body really wants to get something out. The normal flora, these are going to be good bacteria that are going to compete with the microbes. A lot of times they will secrete inhibitive chemicals as well. So with the second line of defense, we have our antimicrobial chemicals and proteins. So this would include the interferons. This is going to be produced by your virally infected lymphocytes, macrophages, and fibroblasts. They'll diffuse into neighboring cells to help induce the synthesis of antiviral proteins. Complement, it's a group of about 20 proteins. They make up about 10% of the globulin part of the serum. There is a cascade of events involved in their activation, and it helps to protect the body by making the MAC, the attack the bacterial membrane complex, that will lead to cytolysis. It can stimulate your macrophages so they can do opsonization to enhance phagocytosis, and then activating inflammation. So we can have iron binding proteins like the transferrins. They're going to inhibit growth of bacteria because they're going to bind up the iron so it's not available to them, basically withholding food. Your antimicrobial proteins, they're short peptides that have broad spectrums of antimicrobial activity. They can lyse bacterial cells, attract dendritic cells, and mast cells that can be part of the immune response. So your interferons, interferon alpha and beta, these cause cells to produce antiviral proteins to help inhibit viral replication. And then the gamma interferon will cause the neutrophils and macrophages to phagocytize the bacteria. So complement is a cascade. Here you can see the complement cascade. First thing, you start with C3. It's going to be activated and split into C3A and C3B. C3B will split into C5, and then that splits into C5A and B. And then the C5B can become C6. That will join with C7, C8, and C9. Or the C5A will go on to stimulate the mast cells. So C3b is going to be the one that causes opsonization. C3a and 5 cause inflammation. And then C5b, C6, C7, C8, C9 all cause cell lysis. So the second line of defense involves antimicrobial cells like your natural killer cells. They're a lymphocyte, but not a B or T lymphocyte. These are able to kill a wide variety of infected body cells, which makes them very useful because they can kill certain tumor cells. They can attack the body by the cell displaying any abnormal or unusual plasma membrane proteins. So they can release perforin that's going to make perforations, and granzyme, which will help to induce apoptosis. The phagocytes, these will include your neutrophils and macrophages. Those come from the monocytes. They'll migrate to the infected areas, and we've got 5 steps to phagocytosis of a microbe. So first we have chemotaxis. This is going to help it be able to identify where the microbe is. Next will be adherence. So it can do this by creating a pseudopod. You can have your lysosome be waiting here. You'll have ingestion where you take it in, and then the lysosome is going to fuse with the vesicle that contains the microbe, and then allow its digestive enzymes to be thrown into that vesicle. So that will lead to step 4 with digestion. And then you eventually have the killing, and then the residual body is left with the indigestible materials. Here's another diagram showing antigen presentation. So you can have the microbe taken in, it gets digested in the phagolysosome, and then part of it is going to be displayed on the surface as a cell is able to now signal additional immune cells. This is an example of a macrophage here. So it literally means big eater. So with the second line of defense, this will involve inflammation. So we've got non-specific defense responses in here that are just responding to the damage in the tissue. So we've got 4 main signs and symptoms that will go with this: redness, heat, pain, and swelling. So you also have altered movement in as the 5th part of the inflammation acronym. The goal of this is to try to dispose of the microbes, keep them from spreading, and then prepare the site to have the tissue repair. So there's 3 main stages to this. First, you have vasodilation and increased blood vessel permeability. This is going to get more blood and tools to the scene. Emigration, your phagocytes are going to migrate and do phagocytosis. And then finally, tissue repair, where the infected cells are going to release the chemicals that will trigger the inflammatory response. So we have multiple chemicals that are going to work at doing inflammation. Histamine is a big one. It's going to cause vasodilation. It will increase the permeability of the blood vessels. The kinins are going to do the same thing. The prostaglandins, they are going to help to control the intensity of the histamine and kinin effect. And then the leukotrienes increase the permeability of the blood vessels and phagocytic attachment. So looking at the process of inflammation, when you increase the diameter of the arterioles, it's going to allow more blood to flow to the area to bring more supplies and help remove the debris. By increasing the permeability, the substances normally retained in the blood are able to pass out as well. This would include antibodies and clotting factors. And then with phagocyte migration and phagocytosis, your neutrophils are going to predominate in the early stages, but they're going to die off quickly. Monocytes are going to then transform into macrophages. They're going to be a little bit more potent than those neutrophils. So when you have pus, it's actually pockets of dead phagocytes in damaged tissue. So here's a couple of terms. Margination is when the phagocytes stick to the endothelium. Diapedesis is when they're going to squeeze between the endothelial cells. So the way I remember it, margination, they're on the margins, which is the endothelium. Diapedesis, petty, like walking, they're going to be able to walk or squeeze between the endothelial cells. So this is showing here with tissue repair. So up the top you have a scab, below that is a blood clot. Below that you would be regenerating the epidermis, and below that regenerating the dermis. So here looking at tissue repair, so you would have the bacteria or pathogens that would enter the wound. Platelets are going to come from the blood release, blood clotting proteins at the site. Mast cells are going to secrete factors that will mediate the vasodilation and vasoconstriction. This is going to get more blood flow in the area. The neutrophils will start to kill and degrade the pathogens. And then they'll start to do phagocytosis. The macrophages will secrete the cytokines, and that's going to attract the immune system cells to the actual site and activate the cells in tissue repair. Then you would finally have the inflammatory response. So a fever actually serves an important purpose. It's an abnormally high body temperature. So your hypothalamus normally has your body temperature set to be at 37 degrees Celsius. But some of the pathogens that can cause the phagocytes to release IL-1 or interleukin-1 can change this. The hypothalamus will release prostaglandins that will reset that hypothalamus to a higher temperature. That's going to tell your body to have a fever. Your body is going to increase the rate of metabolism and shivering, which is going to raise the temperature. With vasodilation and sweating, the body temperature would fall. So when the fever breaks, we can sometimes refer to it as a crisis. So the advantages of a fever: it will help increase the transferins, it increases IL-1 activity, produces interferon. It's also just a less ideal temperature for the microbe. Some of the downsides to having a fever: you can have tachycardia, acidosis, or dehydration, and if it gets too high, 44 to 46 degrees, it can be fatal. So in the next video, we will talk about adaptive immunity, or the third line of defense.